Good morning, class 7. Today I will start the topic of the same chapter, classification, kingdom classification number 1. Today's topic is activities found in amoeba. So it gives us some ideas about the prokaryotes, how they are taking their nutrition, how they are complete their locomotion, and how they are performing their reproduction and each and everything. So children, first I will start by the experiment done on the amoeba, that is the method of taking nutrition in it. So if amoeba is a prokaryotic organism, so in the prokaryotic organism they will engulf their food as in the whole. They are not chewing it because they have no teeth. They are not crush it because they have no gizzard, nothing to crush the food. Simply they are engulfing the full food, animal or full amount of that food into their body, that means into their cell. Yes children, suppose this is the amoeba or it is a, this is the prokaryotic cell. First, when they got their food particle, they start to elongate their pseudopodia around that food particle. After that, these pseudopodia come close to each other. Then in the next step, rest part of the body of amoeba remains same, only the pseudopodia becomes longer and come closer to each other. In the third step, these pseudopodia will join and then they form the complete lining of their body, and food is engulfed into the vacuole known as food vacuole, and in the cell as they have all cell organelle are present without their covering, so surrounding the food particle there will be the collection of lysosomes. Lysosomes are initially they are termed as suicide bag. It means that they can help to digest the organic material into its component, and then these components can be given to the cell. So in the third step, all the lysosomes are present surrounding the food vacuole. And when lysosome totally digests the food particle, then they form a surrounding to it. There is a covering, there is a fine membrane like, and this is known as the food vacuole. After digestion of the food, they form the food vacuole and the nutrient can be given to the cell of the amoeba, and in this way these are the cell organelle, and in the next step these food vacuole come closer to the membrane, and membrane gets ruptured and food vacuole throw the undigested material or the remaining part of the food, they throw it outside, and in this way the amoeba or prokaryotic cell can engulf their nutrition, can digest it, and then can mix into its cytoplasm. So this process is called as phagocytosis. So during nutrition, the prokaryotic cells shows phagocytosis, and phagocytosis, first they engulf the food then by engulfing the food they change it into its component, and this component will be assimilated into their cytoplasm, and in this way the prokaryotes get nutrient into its body. And after engulfing the food, that is ingestion, and after ingestion there will be the digestion done by the lysosome. After digestion there will be the assimilation. When the food vacuole gives the nutrient to the cell of the amoeba, it will be the assimilation. And ingestion means when they come closer to the membrane and membrane becomes rupture, and then they can throw out the remaining part of the food or undigested food material. So this is the total process of phagocytosis. Next, children, it shows the locomotion. How the amoeba or the prokaryotes can complete their movement or locomotion. It is very slow, very slow process, and due to that they are microscopic, so any person can't identify their locomotion. But if it is seen, or if it can be seen under microscope, then we can see that amoeba also can move from one place to another place. Children, if they can take their body from one place to another place, then it is known as movement. So this is locomotion. Now children, here this is the locomotion. This is the basic line or baseline where the amoeba is there. So amoeba, here children, I have drawn here with the help of green color. This is endoplasm and outside to it ectoplasm. So if the amoeba wants to move in any direction, first they dilute their ectoplasm and increase the amount of endoplasm, and in this way they can throw the pseudopodia in the desired direction. In which direction they have to move? They throw the pseudopodia, and along with the pseudopodia they release or they pass the ectoplasm along with the endoplasm, due to which the pseudopodia also gets cytoplasm, and when it gets cytoplasm they also can get the strength about it, and when they have to move in one direction, then they can drag their another part of the body towards the same direction. In this way amoeba can move from one place to another place. And this is A part and B part. Now children, C part. In the C part I have shown here, amoeba wants to move in this direction. So amoeba thrown its pseudopodia towards this direction. It becomes small, pointed, and slowly when they send the endoplasm along with the ectoplasm, then it becomes towards this direction, and at that time the amoeba can drag the part of their body towards in the same direction, and then they can move or they show the locomotion. And this locomotion will be only for that prokaryotes and especially in amoeba. Children, in the prokaryotes or suppose eukaryotes also, they are protista. So in the protista, these amoeba belongs to the protista. They also show the same type, but in the protista, in the other animals just like paramecium, they have cilia. So they will show the ciliary movement. So in this way, some different type of unicellular organism will show the movement with the help of different locomotory organ. One here example it is given for amoeba, that is, it is the protista belongs to protista and belongs to the protista. Those animals have different types of locomotory organ. Just suppose paramecium have cilia, euglena have flagella, amoeba have pseudopodia. In this way they can show the movement, and hydra have tentacles. So these are the movement of prokaryotic cell along with the eukaryotic cell also, because this amoeba belongs to protista. So children, phagocytosis done by the amoeba, that is the process how to engulf their food. Then amoeba show the locomotion by liquefying the endoplasm and ectoplasm. Then they increase the volume. In this, after increasing the volume, they can throw the pseudopodia in the desired direction, and they can move it from one place to move their body from one place to another place. Okay children, for today it is over. God bless you. Thank you.